Okay, so when I say genome editing, everybody knows what genome editing is, or stands for? Gene therapy. So most people are familiar with gene therapy. So I will try to explain a little bit about genome editing in this case, and none of the studies are in clinical trials right now. So we are doing research in mice, and we are trying to figure it out, how to actually treat mice that potentially we can take it to patients. So I was trying to think about how to think about skeletal muscle, and the easiest analogy I could think about, it's a bundle of bundles. So one can think about a muscle cell, it's like a long stretched hose that goes between one bone to another bone. And all of these hoses, they can form a bundle, and then bundles can form bundle of bundles, and that's going to be a muscle. And you can imagine also that this hose is going to be with a like water spout to move water to fill, for example, a pool. So for muscle to stay healthy, what do you need? So all of the health of the muscle is going to depend on this structure called DNA. So DNA is going to build important components for muscle function. If some of these components are not functional, muscle is not going to work properly. And DNA is going to define basically what we are, whether you have blue color eyes or brown color eyes, curly hair or straight hair, and so on. So in DMD, we have mutations in the dystrophin gene. And if you look back, so DNA, those are the letters of the DNA, and sometimes even if one letter is mutated, it can lead to a mutation in DMD. When I ask, most people have mutations that are caused by deletions. It means the whole stretch of DNA is gone. And when I say about duplication, some portions of the DNA are repeated twice. So in a healthy muscle, that's what we are seeing. So all of this, if you like the analogy of garden hose, those are different hoses that are kind of between one part of the bone to another part of the bone. And in case of DMD, its muscle is getting destroyed, and we are seeing a lot of fibrotic tissue and a lot of cells, of the immune cells that infiltrate muscle. And so muscle is not functioning anymore. And in case of Duchenne muscular dystrophy, mutations are caused by mutations are in a DMD gene that lead to the absence of dystrophin protein. So you can also think about DNA as a cookbook. So we have a cookbook, and every cell in our body has the same cookbook. But bone is going to be different from liver, is going to be from muscle, and it depends on what kind of recipes we are going to derived from this cookbook. Cookbook also can be very elaborative. So you can say, if I want to make a cake, I can say, find 3 eggs and mix it with 1 cup of flour. Whereas the recipe, if somebody is going to read it to me, I said, can you do it quickly? So, okay, 3 eggs, 1 cup of flour. It's going to be sufficient to make a cake at the point. So eggs, flour, and cake is a protein that is going to be, in this case, a dystrophin. So if we have a mistake in a DNA, we are not going to make a recipe, it's not going to be right, and we will not be able to build a protein or a cake, and in this case, dystrophin. And that's what basically it is. In our cookbook, if there's a spelling mistake, as I said, one mutation or deletion or a duplication of certain letters, recipe is going to be altered and it will lead to no cake and no dystrophy. So current therapies in Duchenne, and for example, what we heard this morning about NF-kappa B inhibitor, so they are not touching the DMD gene itself. They are helping to maintain a muscle function differently. In the garden hose analogy, it's basically, let's say you have a hole in a hose and you're going to put a masking tape on it. So you're trying to protect it, but eventually it's going to tear off and you have to do something else, something else, something else. When we talk about microdystrophin therapy, and Dr. Mindela is going to talk about it after me, essentially you can think about it as a leaflet in a book. So we are not changing a book, but we are just adding a page. We are adding a page, but it's not bound to it. And if something happens with cells and cells divide, we can lose this leaflet. But microdystrophin recipe is there and it can be read. So microdystrophin will lead to smaller protein, partial cake, but it's still a cake. So we can live with it, we get a prolonged life. And usually, when we think about it, it would be if you take a patient with Duchenne, we can convert patient with Duchenne to a patient with Becker muscular dystrophy. So it's a milder condition. And when people talk about exon skipping therapies, like antisense oligonucleotides, they basically interfere at that stage, on the stage of the recipe. And one way to think about it is like sometimes less is better. Sometimes too many eggs are going to ruin the cake. And let's say, and at this stage, if somebody said, okay, one egg is dropped, we can actually build a partial cake. But problems with this therapy is going to be, they will need to be repeated over and over and over again. So what we are trying to do, we are trying to tinker with the cookbook itself. We are trying to change it on the level of DNA. We are trying to make changes that we can make the full entire cake. And that's basically, we want to convert situation with a dystrophic muscle in a situation with normal muscle. And so red color here that's where dystrophin located, around the cells. So in a garden hose analogy, it's the periphery of the muscle. So now, how do we do it? We are doing it using CRISPR, or genome editing tool. So you can think about CRISPR as molecular scissors. It contains two components. One is a protein called Cas9, and one is a guide. It's an RNA molecule, and it's the search bar. And only when you combine those two components, so the search bar is going to go along the DNA, find portion where it has to cut, where it finds some similar sequence, and only then Cas9 is going to cut. So it's something like that. Find, we can remove portion, and then cell is going to join two pieces together. And for duplication mutations, that's what we are going to do. So duplication mutations, as I mentioned, is basically when portions of DNA are repeated. And in this case, it's a portion from 17 to 30 is repeated. So you can also think about DNA, so it's a jigsaw puzzle, and puzzle has to fit together in order to function properly. Here, on mRNA level, we are having straight line. Everything fits perfectly, it's going to be functional. Here, we come to here, 30 and 18, they don't fit together. It's not going to function, and basically in this case, we are going to we are going to build cake and normal dystrophin protein, and in this case, it's going to be no dystrophin. So, two ways to think about removing duplication. Duplication is something that's repeated twice. So we can find a guide that is going to be cut here at the junction and cut outside, and basically removal with two guides. Similar strategy has been tested for deletion mutations, and that's a pro and cons. It's a two guide requirements. It has to be location specific and specific for each duplication. So we designed an approach that is one guide approach. Because the sequence is duplicated, we can use only one guide to cut it twice. So only one guide, less location restrictions. Same guide can be applied for multiple duplication, and the strategy was not tested. So we had patient cells from a duplication 18 to 30, and we said, okay, can we use CRISPR right now to remove this duplication and restore normal dystrophin production? So in this situation, if you look at the healthy individual, you have dystrophin. Individual with duplication, we have no dystrophin. But no dystrophin. And as soon as we introduce CRISPR, you are going to see that dystrophin starts to appear in treated samples. And as a scientist, that's when you see the most elation. As soon as you're seeing this result, when something was not there, but it was present in the DNA book of the individual, and right now you're starting to see in it, it's an eureka moment. So it actually can work. We can actually take something, non-functional gene, and we can make it functional again. And we said, great, we are going to now to test it in mice. Where do we find this mouse model to test it? There's no mouse model. So we had to actually create our own model to be able to test the strategy. And to test this mouse model, it's another entire lecture on that I can give, because it took a long time and a long of sleepless nights. But at the end, we made the mouse that has duplication from 18 to 30. So now we can say, okay, finally, we made it. Can we now treat it? First of all, of course, we showed that DMD duplication model has no dystrophin expression, so dystrophin is not present, and on a staining, it's obviously dystrophic because we have fibrosis accumulation and infiltration by immune cells there. Okay, model is there. Now let's try to treat it. And the way we treat it, we package our genome editing components in a virus. And those viruses called adeno-associated viruses, or AAVs for short. We can put it there, and we say we are going to treat our mice very early in life, at day 2, and we are going to analyze them at 6 or 14 weeks to understand what's happening. Can we actually now make the treatment work in mice? Here is first example. If you look at the recipe, this recipe is longer and this recipe is shorter. So shorter corresponds to a healthy and longer corresponds to DMD mutation. As soon as we deliver treatment, now we are seeing that we have both. Longer form is still present, but we are seeing appearance of a shorter form. And that is the wild-type, full-length dystrophin recipe is being produced in our mice. Of course, we wanted now to look at whether dystrophin actually protein is being made. And we looked in heart, and we are seeing in a healthy mouse a lot of dystrophin, and we are seeing a lot of fibers that in treated mice. All of these red circles indicate that we have cardiomyocytes now that producing full-length functional dystrophin. Diaphragm, transduction a little bit less, but we are seeing now fibers that have full-length dystrophin. TA, and that's a muscle in the leg of a mouse or hind limb. We are seeing dystrophin. Triceps, like a hand for a mouse, we are seeing a lot of dystrophin. So overall, it looks like the strategy works. At least we can recover functional dystrophin. And now we say, okay, does it help the mouse? Does the mouse now move a little bit better or not? And the way we do it, we put mouse in a box, and we basically going to record how mouse is going to move. And the camera is going to record all of the movements, and then all of the movements, and when mouse is pauses, we can plot and analyze. And if you look at the resting time and compare healthy and duplication mice, you can clearly see that mice with duplication, they rest a lot more. They don't like to walk as much. But treated mice now, they rest less. So it's becoming comparable to the wild-type healthy mice. And the same with total travel distance. If we analyze healthy mice, they travel a lot, they like to explore, they move around. And treated mice don't really like to move that much. And treated mice, they start to explore and move. So actually, this strategy does work. We are able to get a full-length dystrophin in mice, and mice perform better in the functional test. So here, I just wanted to show you this example. So this example is not from DMD. It's from another form of muscular dystrophy that we are working on. It's called LAMA2 deficient muscular dystrophy. But it's just easier to see. So DMD mice, they're not the greatest model because they don't show all of the phenotypes that I see in patients. Whereas here, it becomes a little bit obvious what CRISPR and genome editing can do. So this mouse, 7165, has a disease. So you can clearly see hind limbs paralyzed. We use CRISPR, and that's what happens. So we are able to completely make mice, they have paralyzed and they have skeletal muscle abnormalities. Now they can move around, and they're undistinguishable from wild-type mice. So I wanted to thank all the members from Con Lab, and especially Daria. Daria is the audience today. So she is now research director on Muscular Dystrophy Canada. So she spearheaded this project about removal of duplications. And we have lots of collaborators. And of course, this work would not be possible without support from funding sources, and specifically from Jesse Journey, who found this research. But before I finish, I just wanted to say, okay, it does work in mice. So what's next? What is the future? So what needs to happen for that to go from something that works on the level of animals to actually move into clinic? So first of all, we are following footsteps of gene therapy 1.0. Essentially, people have already worked since 90s to improve delivery vectors. So we can think about what they've done, and we can utilize it. So when I say that we are going to package genome editing component in a virus, we know how to do it because we have learned from them. So it's becoming much easier. Also, in clinic, like right now, there are already clinical trials with microdystrophin and with many other diseases. So they're coming to age. So when the time will come to actually for approval, it's going to be a little bit easier because we are following the footsteps. So that is the issue right now. Because we are changing DNA, something has to also improve, and we have to be cognizant of the fact that by delivering somatic genome editing, we don't change DNA in a germline, because there are a lot of ethical debates about it. So we have to improve our vectors to be able to do that. Off targets. Everybody who you will ask about genome editing is going to say off targets are still a big issue. And what I mean by off target? Because we do cut DNA, we don't want DNA to be cut in some other place. It's like when you put in a GPS, Victoria Street 553, but there could be Victoria Street 553 in London, Ontario, or in Toronto. So we have to make it more specific so that actually we want guide to bring Cas9 only where we want it to be. So that's the area for improvement. Cas9 is a bacterial protein. It's foreign for the body, so immunity against that is going to happen. So we have to find a way to deal with this immune response. So that's an area for improvement. And new delivery methods. So gene therapy is built on using adeno-associated viruses right now as delivery vectors, because they do work and they get into the tissue where you want them to go, more or less. But they also persist in tissue for a long time. It's great for gene therapy. It is not so great for genome editing. Ideal delivery vehicles are going to be, you come there, you cut, you change, and it's gone. So we are working and we are collaborating with our partners from U of T about designing biodegradable nanoparticles to deliver genome editing components. And of course, idea here is that maybe in 10, 15 years, maybe less, a doctor in the office will be able to prescribe. I'm going to prescribe CRISPR, and I'm going to prescribe that guide, and it's going to be actually injected in patients. That's our goal. Thank you.